Hello, this is Evan Gudell, and this is my research project I've done with Dr. Callahan for the past 2.5 years, and it is Saccharomyces cerevisiae cell wall isolates are potential inhibitors of bacterial growth. And so what I do is I look at this binding interaction that occurs between type 1 fimbriae of pathogenic bacteria and mannose-sugars. And these mannose-sugars are expressed on the surface of our intestines. They provide an adherence point or an anchoring point for these pathogenic bacteria to infect you and prevent being washed out of the intestines. However, these mannose-sugars are also highly abundant within the cell walls of yeast. And so the thought is, if I introduce yeast between these fimbriae and the mannose-sugars of the intestines, could it prevent adhesion and infection, or could it even displace bacteria from the intestines? So these mannose-sugars are linked to mannoproteins within the cell walls, and the mannoproteins are highly abundant within the yeast cell walls. And they also have large amounts of mannose. Most importantly, they're linked to this beta-glucan, which provides an anchoring skeletal structure that retains the structure of the cell walls. And so this makes it easy for me to simply separate out the cell wall using autolysis and collect these cell wall husks for my binding assays. Now, obviously, after I collect these husks, I have to make sure, do I have mannoproteins there that are able to bind to the fimbriae of bacteria? And we do see here that we have large amounts of mannoproteins in this overlaid SDS page and Western blot. Most importantly, though, the mannoproteins are retained in the cell wall, as when we simply just run the cell walls on an SDS page or a Western blot, we don't see much of anything. But if we treat that with zymolyase first and I degrade the beta-glucan linkages, it frees up mannoproteins that are then able to be visualized. Now, the real most important part of my project is the binding assay. And this occurs with the undigested cell walls of the yeast, as I'd like to preserve the 3D structure. And what I do is I take a 96 well plate and I coat it with my cell wall treatments or various controls, such as free mannose-sugars or general proteins. And I then introduce an E. coli culture, let it adhere to the plate, and wash it away, followed by overlaying with fresh media. After this, I take readings of an OD 600 every 30 minutes to an hour for 16 hours. And the thought is, if we have more binding interaction occurring between the bacteria and my cell wall treatments compared to the controls, then less of them will be washed away in my initial steps and I'll retain more initial bacteria. And this will allow us to enter into an observable range much quicker than something that would be an uncoated 96 well portion. So this is one of my binding assay results. I've run it 4 or 5 times now, but it does take 16 hours. And unfortunately, we do not see any significant difference between the treatments of my cell walls or the free mannose sugars or general proteins. Now, this is unfortunate, but it is not totally unexpected, as my collection procedures of the cell walls could have possibly disrupted them to the degree where the binding will no longer work. Another big point is that the type 1 fimbriae of the bacteria are a pathogenic feature of them. And so the strains we are able to work with at Fisher are generally safer and may not express as much of these type 1 fimbriae. So they may not display as high of a binding activity as we would like. That would make it easier to look between the different treatments. In the future, I would have liked to look at this more and check how my collection procedures affect the binding activity. And I did actually buy a different bacterial strain that has a higher expression of these type 1 fimbriae while still being safe to work with. Unfortunately, we never quite got the chance to use that. And the big question I would have liked to look at and answer is, could these treatments, if the binding activity is strong and if it occurs well, could they prevent adhesion, or could they remediate adhesion of the bacteria to the gut? Could they displace them and take them out and treat an infection?